Hi class. Today I'm presenting my presentation on the enzyme Ketopantoate Reductase, also known as KPR, and the mutagenesis of that. So we're focusing on staph, like staph infection, Staphylococcus aureus, which is a bacteria that has resistant strains. That causes problems in medicine when we need antibiotics to prevent these infections and they're resistant to it. So the main idea behind this project is to create a mutation that will eventually result in the production of a protein that will be able to fight against these resistant strains of staph. Right here, I have an overview. I have a flow chart that I've made on the whole project we've been doing. So as you can see, up here, first we're going to examine the structure of this of this enzyme through UCSF Chimera, which is a program that I'll talk about later, but it helps us identify the structure, and then we're going to use that to hypothesize different mutations around the ketopantoate protein that could affect the functionality overall. So we're going to do that by designing primers. Those primers we design through STM Assist and SnapGene will help us create a mutation that could ultimately negatively affect the function of this overall process, which would benefit us in the fight against the staph bacteria. So moving on, just the overview, we're going to create LB agar plates. We're going to run PCR mutagenesis. We'll talk about that later. We're going to run an agarose gel and run gel electrophoresis to confirm our PCR results. After that, and we've confirmed our plasmid is in the plates and the colonies and the bacteria, we're going to transform that actually into the bacteria itself. We'll talk about this later, but then we'll purify it. We'll do another transformation, and then we'll get our protein expressed at a larger level. But that's just a simple overview. We'll go ahead and talk about it more. All right, KPR, the enzyme, specifically with Staph aureus, it's a key enzyme. As you can see, it's a key enzyme in the pathway for metabolism. You see, this enzyme right here, it's shown via Chimera, but this enzyme KPR will reduce ketopantoate. It's an NADPH reduction of ketopantoate to pantoate. And then pantoate will be transformed into pantothenic acid, which is used to create coenzyme A, which is a key process in metabolism. Every organism on the planet needs coenzyme A to survive. So that's why you could see, if we find a mutation in this, how important it can be to disrupt the functionality of the whole overall process, which could benefit us in many ways. But another key point is when looking at Chimera, we notice that Staph aureus is a dimer. Rather than in E. coli, it's a monomer. So that means there's two identical halves of this thing, of the structure, that are connected. So we know if we could find a way to disrupt that connection between the two halves, we could figure out a way to better our understanding, which would help us create a mutation that could help us against the fight of antibiotic resistant bacteria. Okay, let's talk about our experimental procedure. So this is site-directed mutagenesis. Site-directed mutagenesis is pretty much we are going to look at a specific spot on the DNA sequence that we want to mutate, and we want to change it from one amino acid to another, which could change the functionality of the whole molecule or organism. And we know this because structure, so often structure is the basis of functionality. If you change the structure of something to an extent, it changes the functionality. So we understand, see right here, for example, our project I'm doing and I'm conducting right now, we have these two main spots at 194 glutamate and 279 phenylalanine. And I've chosen to mutate this phenylalanine at 279 to aspartic acid. And that difference alone, we'll talk about that later. This is just an overview, but that alone could cause disruption in the functionality of the whole system. So we're just looking about what we do know, what we don't know. The information gap is, we know a lot about the E. coli, KPR and E. coli, but we want to learn more about KPR in Staph aureus. And as you can see, we're gonna play around. We're gonna make a couple mutations. I'm gonna focus on this phenylalanine to aspartic acid mutation. But by that process, by mutating and seeing how the overall system reacts, is how we can understand what we need to do in order to fight these bacterial infections. This is just another continuation. So we know we're going to use Chimera to see the overall structure analysis. PCR. With PCR, we will create a mutant plasmid that we've chosen, such as my phenylalanine to aspartic acid. So we will, through Chimera, we'll pick a mutation, we'll implement that mutation through PCR. Once we verify that through a gel that we have our mutation, we will add that mutation, we'll transform it into E. coli, and then we'll plate it on kinomycin antibiotic plates. And we will have a gene implemented in our plasmid. We'll have a gene implemented in our plasmid that will encode for the resistance to kinnomycin. So when we plate it, we'll know if we have colonies growing, it should be our mutant plasmid, because our mutant plasmid is resistant to that kinnomycin. And if the bacteria doesn't intake our mutant plasmid, it shouldn't show up because the antibiotic should kill it off. And this is when we get to blue-white screening, which is when we plate it, we'll add a specific reagent that our mutant plasmid will show up blue on the plate, while the other ones show up white. It's just a technique used throughout lab, but we'll talk about that later. Okay, back to the first step. Our first step is to go through UCSF Chimera, a program on the computer that will help us identify all surrounding molecules as well as our main molecule we're focusing on, the protein ketopantoate. So ketopantoate is the main protein, and as you can see, we have all these surrounding amino acids and molecules around it. So we just want to know, if we mutate one of these, which one, or if many, and what could they be mutated to change and disrupt the entire function. So we know this ketopantoate is reduced to pantoate in the biosynthetic pathway in this bacteria, which creates pantothenic acid, I've already talked about this, which is then converted to CoA. This is very important because bacteria does this, well, they have this pathway, but mammals like us, we don't rely on this pathway. We get vitamin B from our diet. So that's why this is so important. And if we have the correct mutation, the medicine wouldn't affect us because we don't need that pathway because we get it from our diet. That's the main concept behind this. But after we find a mutation, such as my phenylalanine to aspartic acid, we will design primers. This is used with SDM assist and SnapGene and coincide, but we'll design primers through SDM assist. We'll have a basis sequence that we'll add to it, and then we can go ahead and pick the exact mutation we want. Once we pick that mutation, it'll send another, we can use that link, upload it to SnapGene, and it'll create, let me go ahead and skip forward, it'll create this plasmid on a close-up level so we can identify what we want. And as you see here, we have our reverse primer, forward primer. These are the primers that are going to create our exact mutation that we are looking for. Let me go back. So before that, if you don't know what plasmids are, so bacteria has DNA, but they also have plasmids, which is circular DNA, and we're going to use these plasmids because it's easier to get these plasmids in rather than the whole bacterial DNA. They're easy circular strands that we can get through the cell membrane and get them expressed. They're the genes expressed that we want. And we're going to use restriction enzyme sites. These are the sites that get cut during, let's see, like right here, like the restriction enzyme site I have in the mutated plasmid is XBAI, and this restriction enzyme site will cut right here. So when we run a gel, we should see a linear, instead of this circular plasmid, we should see a linear, because there's a cut right here. It'll just be a linear, won't connect. And we'll be able to identify that through our gel. But as you see here, these are the primers. These are the exact primers I had, and they have these little mismatches right here, which cause the mutation. They change. It's a silent mutation, so we can verify mutagenesis afterward. All right. See, exactly. Restriction enzyme sites can be predicted in gel electrophoresis banding patterns. Let's go ahead and move forward. So PCR mutagenesis. As I've said before, phenylalanine at the location 279 on the DNA sequence to aspartic acid. And I chose that because phenylalanine is nonpolar, and I figured, if we change this to a polar molecule, we mutate it to a polar molecule, that should have clear functionality issues. That should change the function, and we should see something there. This is just a picture of the pipetting tools we use for adding reagents and mixing and all that good stuff, and that's at micromillimeter level and microliter level. So yeah, gel electrophoresis, that's how we verify our PCR, and then band analysis. So once we have our PCR, we will run it through gel electrophoresis, and we'll be able to see exactly what we have. Let me explain. This is how we do this. So we create this gel, and we have this piece in it that creates these slots. And these slots, we will inject our sample. Like for example, right here, we eject our sample right here, put it on a electrode, and then it once the let's see. Let me explain this first. The DNA ladder, we add it in the first slot. I always do it in the first slot just so we can know where it's at. But the DNA ladder is pretty much like it could be like a key in a map. At a certain point, we have, like, these are throughout each DNA ladder. So each band, so say it's like the third band, that could correlate with 6 kilobytes or whatever. It just, the key concept you want to learn from this is that each band, this helps us determine the size of the pieces of DNA and plasmids that we have. So example, right here, this is my PCR sample, and you can see a bright band right here, right around the 6,000 kilobase length, or 6 kilobase, 6,000 base length, same thing. Either way, but we know that because right here and right here it lines up. So we see, okay, what is the third banding down from a DNA ladder, this particular DNA ladder mean? And we know that means 6,000, which we expect to be the length of our plasmid. Therefore, we see clear indication of our plasmid there. Does that make sense? Just to throw that in again. So we have the DNA ladder, we have our sample, we get banding from it, and usually primers will be lower down here. But like, this reads what our plasmid is. Let me go back. So this whole thing, we know, is about 6,000, if you can see that, 6,000 bases. That's how the length of it. So this is how we determine what length we have. So if we have that indication of 6,000, which is labeled here, we know that right here is about 6,000, then boom, our sample's there, our plasmid's there, we know it's there. That's just the basis of that. We verified that it's there. All right, and then after we can verify that, we go through transformation. Transformation is when we take the mutant plasmid that we have, that we verified in our PCR, and we transform it into bacteria. Pretty well, specifically E. coli. So we do this by heat shock procedure. So we take our PCR, we add it, we mix it with some E. coli, and we perform heat shock procedure, and we do this by putting it in 42 degrees bath water for 45 seconds, and then directly into ice, directly after. And the change in temperature allows for the permeability of the cell membrane, and it allows our plasmid to enter the bacteria. So that's how we get our plasmid into the bacteria we want. And once that happens, boom, that's all we do. And we incubate it, a shaking incubation with rich media. So we incubate our mixture, which should have some bacteria and our plasmid in that. So we put it in incubator at certain degrees, warm temperature, ideal temperature, with rich media that they can grow. So they'll grow and grow, and this is where the antibiotic resistant gene from the plasmid comes into play, and that'll be created. And we shake it for oxygen. We shake it so there's oxygen, so that increases growth and rich media that they can feed on. So once that happens and they're growing, what do we do next? Well, after that, we're going to plate them. Let me go back. After the transformation, we're going to plate them. And we're going to plate them on right here. We're going to plate them on, if you just focus right here, we plate them on these plates that we have with clindamycin antibiotic included. So these plates, which have our antibiotic on it, they'll tell us what these bacterial colonies have. Well, let me back up. We're going to take the transformation, we're going to add it to these clindamycin antibiotic plates, and we're going to wait for a day or two, come back and see what we have. If we have colonies growing, we understand that, well, our plasmid is there, but we can't assume it. First of all, let me get that. We can't assume it. Once we have growing colonies, we know we know a couple things. It could be a perfect, if we have colonies growing, it could be perfect. We could have our mutant plasmid into the bacteria and it's growing, and the antibiotic resistant gene, which is in our mutant plasmid, is in these bacteria. But we can't assume that. Why can we not assume that? Because there could be wild type plasmids or multiple mutations within these within the bacteria that would allow them to survive in a kinemycin plate. So we have to make sure. That's what you want to do in lab settings. You always want to make sure you have the correct results and you want to verify. So to do this, we are going to, let's see. We are going to do protein purification right there. We are going to select a colony. We're going to add it. We're going to add buffers to it. We're going to make sure the antibiotic's there. So it's so the selection maintains. We are going to, well, another way we could do this, you do centrifugation, which is when you spin it, you put it in a tube and you spin it really fast, and then a pellet of cells will accumulate on one side, and then you're going to add buffers. And when you add buffers to it, it's going to break down all the material that you don't want, because we just want to focus on the plasmid. So it's going to add all the materials you don't want, subtract them. It's going to subtract all the things you don't want, cell walls, all that. All we want is the plasmid. So once we have the plasmid only purified, after we add buffers and all this, or another way we could do it is do isopropanol precipitation, which is alcohol, salt, pretty much does the same thing. So once we have only the plasmid, we're going to send it off for DNA sequencing. This is how, once we have it DNA sequenced, we can know exactly what's in it, and we can verify our mutation is present. And let's see. What next? Protein purification, yeah. All right, protein expression. So once we verified that our mutation is there, we're going to go to protein expression. We want our protein to develop and to grow and to multiply. And to do this, we're going to add a chemical. We're going to do another transformation. We're going to do a transformation number 2. And this transformation is similar to the first transformation we did, but the difference is we already know that our mutant plasmid is there. So once we can verify our mutant plasmid is there, we're going to do another transformation, except we're going to add a chemical, IPTG. This chemical works as an on switch for protein expression. So once it's added, the protein is just going to start making and making and making. The gene encoded is going to start making proteins. And the bacteria is going to start making proteins. And after that, we can verify it, and we know, okay, here's our protein that we've been developing. We can test it, verify that it's working, and that it's the exact thing we need. I just wanted to throw this back in there just for questions and stuff like that. But this is the overall process. We identify the structure. We figure out, okay, what could affect the functionality of KPR? What mutation do we need to implement? We go to SNAP gene and SDM assist to aid in designing primers that will craft this mutation. Once we do that, we make LB agar plates, and then we run PCR mutagenesis. When we do PCR mutagenesis, we get the exact mutation we want. It's a process of adding a bunch of reagents, incubating. But to simplify it, we're going to do PCR. We're going to run a gel, transform it, purify that transformation to make sure it's there, make sure it's the exact plasmid we want. We're going to do a transformation again. This time we're going to add that chemical, which acts as an on switch, and it's going to It's going to induce protein expression. Protein induction is going to, which is when proteins are made at a high rate, which is what we want. Okay, I know it's a lot of information. Where do we go from here? Well, right here, we're going to finish the project. As of right now, we're only at the transformation step. We transformed, but when plating the colonies, we didn't have the colony appearance that we wanted. So we're going to have to redo that. There could be pipetting errors. Nothing's always perfect in the lab setting, but that's why we are persistent and we work hard. We're going to gain results from the projects, and we're going to ultimately help with medicine and send it off to companies so that medicines can be produced to fight against this staph resistant bacteria. And that's our main goal. We want to help people. Thousands of people are killed each year from this, and antibiotic resistant bacteria is a major problem, and this is just one experiment that could help in that. And yeah, after all is said and done, I'm going to continue to work hard. Thank y'all for giving me your time, and I hope y'all have a good day.